Hello, everyone. Welcome to another MaxWell webinar. My name is Obien, and I will be your host for today. Thank you very much for connecting to us today from all over the world. And this is the second webinar that we have for 2021. Okay, so let's begin. For today's webinar, we have two speakers. The first one from MaxWell Biosystems, our in-house expert in retina and organoids, Dr. Szilárd Sajgó. He is an R&D scientist. And our main speaker, our guest speaker, is Martina Gennaro. She's currently a doctoral researcher at the Institute of Molecular and Clinical Ophthalmology in Basel, or IOB, under the supervision of Professor Boton Roshka. And she's also a co-author of a recent Cell paper that we will also feature in today's talk. And yes, so maybe next slide. Thank you. So this is the outline for today's webinar. First, we will have an introduction by Szilárd about MaxWell Biosystems and about the applications of our products to the retina research. And next, we'll have the presentation by Martina on morphological, functional, and transcriptomic correlation of retinal organoids to the human retina. So this is really a very high impact work. And this is the first time that retinal organoids has been characterized in terms of morphology, function, and transcriptomic and its correlation to the human retina. All right, so maybe before we begin, please make sure if you have any questions, you can write them here in the chat. And we also have links every once in a while connecting to, for example, content related to the webinar that we have right now, also here in the chat room. So at the end, we will go to our Q&A session, wherein we will read some of your questions here in the chat. And then our guest speaker, Martina, and also our in-house expert, Szilárd, will also be able to answer these. So anytime you can write in your questions. All right, so let's begin. Hi, Szilárd.
Hello, everyone. Hi, Marie, and welcome to our latest webinar. And let me just start out by telling you all on what MaxWell Biosystems does and who we are. So here at MaxWell Biosystems, our mission is to provide electrophysiology platforms and systems and also solutions to advance scientific discovery and to help scientists and companies to accelerate the development of novel drugs. Our core expertise lies in different aspects of microelectrode array technology, including but not limiting to hardware and software development. And our customers are from academia, the biotech industry, the pharmaceutical industry, and also different contract research organizations. So what is actually our setup and what do we measure with it? To start off, this is an electrophysiology tool, but unlike in the case of patch clamp where we record intracellular signals, our sensors are located outside of the cells, thus we record extracellular action potentials. With the application of these high-density arrays, we have multiple of these small sensors called electrodes at super high density. Thus, we can detect signals from one single neuron along multiple points, for instance, along their axons, their axonal initial segments, dendrites, and so on. So now, what are the advantages of using these high-density microelectrode arrays? While in conventional MEA systems, cells between electrodes are not detectable, and multiple cells on one electrode only allows for the measurement of average signals, using the MaxWell HD-MEA, it allows you to have an electrode in the correct position, thus you can record every single cell in your culture. And due to the ultra-small sized electrodes, you are always close to your signal source, and they're able to record from, for instance, from small axonal potentials also. And as you've seen the beautiful cellular fireworks image here on the right, this highlights the resolutional capabilities of our system. So just to highlight once more. Now, what are our products? So we have two major products currently on the market. We have Max 1 and Max 2. MaxWell is our single well format, where you only have one chip, mostly suitable for acute applications, but also tissue culture. And then we have the multi-well framework of Max2. This uses 6-well or 24-well formatted chips. And this mainframe is also a tissue culture incubator that controls the temperature, the humidity, and the CO2 concentration, so you can maintain your cultures while you're doing your long-term recordings. Both systems use the same chip architecture with 26,400 platinum black-coated microelectrodes. They both have an 8mm scale recording area per chip. They have very low noise readouts, and the electrodes are not only capable of recording electrical signals, but they're also capable of electrically stimulating. Now, central to our system is of course, our software that is called MaxLab Live, that enables the accurate visualization analysis of the data. And aside from visualization, MaxLab Live features a user-friendly assay to access and analyze your neuronal activities. And we have 3 core assays called activity scan, network scan, and axon tracking. And just to briefly describe this, so in the activity scan, what you can do is you can define where your neurons are on your array, and you can also define the level of the activity of your neurons. So this is very similar to a microscope image, where you can not only define where your cells are and potentially what the morphology of your cells is, but also what is their electrical activity. Now, once you have identified where your cells are on the array, you can continuously record them, and you can perform a network connectivity assay on them, where you can answer different questions, whether your neurons are interconnected, whether they are firing synchronously, and whether there's any oscillatory activity in your neuronal cultures. Now, the next assay is called the axon tracking assay, and this really allows you to zoom in to individual cells, answering the questions: are the axons of your cells developing correctly? Are they functional? Are they healthy? And potentially how fast their axonal propagation velocity is. These 3 assays can be applied for neuronal studies of iPSCs, brain organoids, brain slices, and even retinas. However, the system is also suitable for recording muscle cells, cardiomyocytes, beta cells, and potentially any other electrogenic cell type. And today, I will discuss in a bit more detail on how one would use this system to study the retina and, for instance, retinal organoids. And what would you need before you can start such an experiment? Well, you would need, of course, different hardware and software components. You would need an assay to project different stimulation patterns onto your retina. You would need the MaxWell tissue holder to flatten your retina in the array. And then once you have the flattened retina perfused and temperature acclimated in your system, you can actually record its activity. But what are you actually recording? You are recording 2 types of signals from the retina. So once you shine light onto your retina preparation, you can record microERG, which is basically a local field potential, and you can also record action potentials. Now let's discuss these in a bit more detail. So what is this microelectroretograms or microERGs? So in large scale, these are composed of the sum of electrical activity, electrical potential changes in different retinal cell types. And it has, in general, 3 major components. It has an A-wave component that is originating predominantly in rod photoreceptors. It has a B-wave component, which originates in bipolar cells and neuroglia cells. And it has a slower C-wave component originating from retinal pigment epithelium. Now, these ERG signals can be very useful when studying, for instance, different mutations or different diseases, because they can really tell you at what level your retina is affected in your disease of interest. And then, of course, you can also pharmacogenically alter these signals and modify them. Now you can also record, of course, as I mentioned, action potentials. And by recording action potentials of the retinal ganglion cells and analyzing them, what you can identify is what is the functional subtype of your cell. Is it a direction selective cell that you have recorded? What is its receptive field? How many photoreceptors are in this receptive field and how wide it is? What is the ganglion cell's light response temporal properties? And of course, you can also track its axons and its axonal velocity conductance. Now, why would you want to study retina using MEA? So globally, the number of people of basically all ages that have some sort of visual impairment is estimated around 300 million. And out of these, approximately 40 million people are blind. Now, I would say luckily, in quotations, but luckily most of these diseases like Leber congenital nystagmus or retinitis pigmentosa or corneal dystrophies or even macular degenerations are diseases that affect the outer retina and not the central nervous system projecting cells of the inner retina. Thus, this really opens up the way for iPSC and organoid-based therapies in the future. Now, ever since 2011, since Yoshiki Sasai demonstrated how to reprogram cells and develop retinal organoids, the scientific community is perfecting this technology with really a huge hope in using it for future therapeutic purposes. Like in the case study highlighted here from Hernandez et al., where they can take iPSC from patients, healthy patients and disease patients, they can reprogram them, they can develop organoids, they can take these organoids, they can transfect, for instance, a red sensitive channel into them, then they can dissociate them, they can transplant them in blind mouse models, and then they can use MEAs to test the retinas of these blind mice in order to see whether light response properties were restored or not. Now, another case presented today in detail by Martina will really highlight the importance of the technology and will highlight why it's necessary to study these organoids in both anatomic, physiologic, and gene expression profile ways. And with this, I will welcome Martina, and I will ask her to share her slide.
Hello, good evening, everybody. I'll just go ahead and share my screen. Okay. So good evening, everyone. Thank you, Mary, and thank you, Szilárd, for the introduction. And thank you, MaxWell, for inviting me to today's webinar. Today's talk will be about retinal organoids and how they resemble human retinas on a morphological, transcriptomic, and functional level. So as mentioned, the talk is based on this recent paper, which was published on Cell from the Roska lab. Professor Boton Roska is here in the picture. He is the head of the laboratory and also the director of the Institute of Molecular and Clinical Ophthalmology, Basel. This study is quite a complex one, which features a variety of techniques. And it has seen, obviously, the contribution of many different scientists here listed, which I would like to acknowledge, and who are all coordinated by the two first authors of the paper, which are Dr. Cameron Cowan and Dr. Magdalena Renner, also here in the picture. So let's start with a brief introduction regarding the organ of interest of today's talk, which is indeed the retina. As Szilárd mentioned, the human retina is the region of the eye that allows us to detect visual signals and also to pre-process this information and to send it out to the brain. The retina is composed of different cell types, as shown in this nice drawing, and it can compute different aspects of vision depending on the region and the cell population. For example, for what concerns the human retina, the center of the retina drives high spatial acuity vision, while the periphery has a lower spatial acuity power and is mainly responsible for night vision. The neural cells of the retina are anatomically organized into 5 layers, of which 3 contain the cell bodies or soma, and 2 layers contain mainly the synaptic connection between these cells. So for example, if we look at this immunostaining, the three nuclear layers are quite easily recognizable, are the ones in gray, while the ones in between are where the synapses occur. Of the three nuclear layers, we can distinguish the outer nuclear layer, which hosts mainly photoreceptors, rods, and cones. Those are the cells that are able to detect photons, so light. In the inner nuclear layer, we have bipolar amacrine and horizontal cells. And finally, in the ganglion cell layer, we have the ganglion cells, which are those which axons will project to higher visual centers in the brain. In the lab, we generate retinal organoids. So a question could be, what are organoids? Organoids are 3D cellular aggregates which recapitulate an organ, in our case, the human retina. The recapitulation occurs on different levels, as we will discover today. And they originate from pluripotent stem cells, in our case, human pluripotent stem cells, which develop in these very nice 3D structures and are more or less 3 to 4 millimeters in diameters. As mentioned by Szilárd, organoids have become more and more interesting in the recent years for their therapeutic and clinical relevance. So to go a little bit more in depth of that, as mentioned, the organoids originate from pluripotent stem cells. A stem cell is characterized by two main features. Its self-renewal power, meaning very simply that a stem cell can give origin to other stem cells, and its pluripotency, meaning that a stem cell can also give origin to many different kinds of specialized cells. So a source of these pluripotent stem cells could be simply a skin biopsy sample from a patient. In fact, those cells can be reprogrammed to have those called induced pluripotent stem cells, which then can be cultured to give origin to different specialized cells. In our case, for example, retinal cells. Those cells can be used for basic research studies or translational or engineering studies. And if they were to be transplanted back in the donor, this would allow a higher immunotolerance. Regarding basic research, an organoid which would mimic correctly a human organ would be a very important tool for basic research, genetical engineering, translational studies, for drug screening, and to have disease models in a dish, for example, and to have patient-tailored research. Obviously, to be used as such a model, an organoid must recapitulate the main feature and the cell types of their target organ, so in our case, the human retina. And for this reason, one of our main question, or the main question, was to what extent do retinal organoids recapitulate the human retina? So going directly to some of our main results, we firstly wanted to have a look at those retinal organoids and see if they would have this multi-layered structure as seen in the adult retina. So as we can see in this direct comparison of an immunostaining, as I described before, the adult retina has these 5 layers, and actually in our retinal organoids, the 5 layers are also detected. And maybe it's also observable in this cross-section of an organoid, where it is quite clear that there is a multi-layered structure. Further stainings experiments show that the cell types, the different cell types that are present in the adult retina are also present and correctly layered in the organoids. In fact, in the outer nuclear layers, we have the rods and cones, which are mainly present in the outer nuclear layer, also in the organoid. And for what concerns the inner nuclear layers, we were able to stain for horizontal stains, horizontal cells, bipolar cells, and star-bust amacrine cells. So our morphology studies showed a multi-layered structure and different cell types. Also, very interestingly, the photoreceptors that we were able to see in the organoids were showing these hair-like structure that are mostly observable in this cross-section, which are the outer segments of the photoreceptors. These segments are crucial for the detection of light. Therefore, we wanted to understand if our organoids were also light sensitive, meaning if the light would be detected by the photoreceptors, and also if this information would then be sent to the other cells. To do so, we expressed the calcium sensor GCAMP6S under the control of a general promoter, EF1 alpha, through an adeno-associated viral vector. The general promoter allows us to target many different cell types, as shown here. So we have cones, horizontal cells, amacrine cells, and we were very careful to focus our experiments on optical cross sections, so that all the layers of the retinal organoid would be under the same circumstances during experiments. So to visually stimulate the photoreceptors, we used the 2-photon laser, and we performed some flashes of 2-photon lasers on top of our organoid. After the photoreceptors were showing some adaptation to this stimulation, we also performed continuous imaging, so to measure the background activity as a control and be able to compare the background activity to the activity during the light stimulation. Here we can see the traces of the activity during background and during stimulation of the outer nuclear layer, so basically the photoreceptors activity, and the inner nuclear layer and ganglion cell layer, meaning all the second and third order cells of the retina. The results were actually very interesting. So the darker gray bars concern the photoreceptors activity, and during light stimulation, the percentage of cells that were responding was definitely higher and significantly higher than the cells active during background recordings. This was also the case for cells of second and third order, so horizontal amacrine cells, bipolar cells. This outcome was showing us that those cells are actually light sensitive. But we also wanted to understand if a circuit was ongoing, or if those cells were spontaneously reacting to light. So to do that, we decided to apply a cocktail of drugs or pharmaceutical compounds, which would disrupt the synaptic transmission between those cells. We repeated exactly the same kind of experiments, but with these drugs in the perfusion system. When we went to measure, we noticed that all the photoreceptors, so all the cells in the outer nuclear layer, were still showing quite a significant difference of activity during the light stimulation and compared to the background, meaning that the photoreceptors were still reacting to light, even though there was a disruption in synaptic transmission. But this was not the case for second and third order cells, which should react to light just because they receive the signal through the circuit. And so this result was actually showing that a disruption of synaptic transmission was also disrupting the activity or the light responsiveness of second and third order cells. Given that our retinal organoids were showing multi-layered structure and were showing all the different cell types, and those cell types were also light responsive, we wanted to investigate how those organoids were developing. And so at which age or week after generation, the different cell types would start arising. To do so, we performed single cell sequencing. To detect the different cell types, we checked the expression of markers, well-known genetic markers. This is shown in these heat or high density maps. For example, we used a couple different markers for identification of mitotic cells, or a couple of different markers for rods, a couple of different markers for cones, and so on. And at the end, we were able to generate a map like this one, where each dot is the transcriptome of a single cell. And the cells were clustered in an area of this map, depending on how similar the transcriptomes were to one another. We then repeated the sequencing at different time points after generation. So we picked some of the most important time points, and we noticed and observed with these heat maps that different cell types would originate at different time points, and that this would stabilize around week 30 to 38. So we determined that organoids that are above week 30 after generation are considered mature or developed organoids. At this point, we were also interested about the rate of development. And to study this, we wanted to compare the rate of development of our organoids to the rate of development of a human retina. We then combined all the single cell transcriptomes of the organoids and compared it to published bulk transcriptomes of human retina during the development, so between week 7 and 20 of development. To compare, we also created a linear model which would predict the transcriptomes, the different ages of the retinas based on the data based on the transcriptomes. So we validated this linear model on the data which was published about human retina, and then we also applied it to the data we had about our organoids. And what we discovered and what we observed, and we're very happy to observe, is that the developmental rate is actually very similar. In the red, we can see the rate of development of an organoid, and the black dots are the rate of development of a human retina. Also, we checked for reproducibility, checking and repeating this experiment for single organoids of different batches, which gave us a very repeatable result. Given that we now had all of these single-cell transcriptomes and data from organoids, we wanted to compare these to single-cell transcriptomes of human retinas. Human retinas can be obtained by donors. So these human retina explants will have to undergo the surgery and have to be transported to the lab. Usually, this is a very long procedure, and unfortunately, the retina's explants will be under ischemic conditions once they are processed for experiments. So ischemia would change completely the transcriptome, and we didn't want to compare an ischemic human retina explants to our organoids. We were able to establish the protocol that allows us to perform a rapid surgery and a rapid transportation of the tissue into a perfused and oxygenated media. And this allowed us to avoid ischemia. We tested this by performing electrophysiology recordings on these explants to observe if the cells of this retina were still spiking, and so they were active, and also if they were responding to light. So not only spontaneous activity, but also responses to light. In parallel, other regions of this retina would undergo single-cell sequencing. So to perform these electrophysiological recordings, we chose multi-electrode array, the multi-electrode array technique. And to perform the light stimulation, we showed this stimulus, which is a very famous stimulation called chirp sweep, which would allow to give different kind of frequencies, different kind of intensity steps, and contrast steps, showing this to the retina. We repeated these experiments multiple times in order for it to be repeatable. And in fact, here, for example, one box would be one cell, and the different lines would be the repetitions of the experiments. Based on the responses of these ganglion cells, and by the way, over 80% of the ganglion cells we were able to record from were light responsive, which is a very good, a very nice result. Given their footprint or how they were responding to light, we were also able to cluster and identify functionally different ganglion cell types. So we were able to detect 5 classes, depending on their reaction to onset or offset, and their response, if it was transient or sustained. So we were able to detect on cells, off cells, on off cells, which was a very interesting and important result. At this point, we had light responsive human retinas, and we performed the single cell sequencing. We compared we compared the single cell sequencing of the peripheral retina, of the foveal retina or central retina, and of the organoids. We decided to do this difference between periphery and central or foveal retina because, as I mentioned in the introduction, different areas of the retina will contribute differently to vision, to aspects of vision. So from these experiments, we also developed an atlas, which is available online to everybody, and which contains all the sequencing results, the count tables, and that will probably be linked now in the chat. So once we had all the data of the single cell transcriptomic, we clustered together all the cells that were transcriptomically similar, and we are visualizing them with these UMAP manifolds. And we were able to detect all the different cell types, but we wanted to also validate that what we were clustering based on the transcriptomes was actually the cell types or cell classes that we thought. So to do that, we compared the cell types to known markers. So here in these maps, we are comparing the transcriptome-based clusters to the marker-defined cell classes. We performed the comparison thanks to an uncertainty coefficient. This uncertainty coefficient was basically evaluating how well the marker-defined cell types were explaining the variability of the transcriptome-based cell types. We also validated this uncertainty coefficient by randomly shuffling the cluster labels, the transcriptomic cluster labels. And once this was evaluated, we were able to measure that there is a high correspondence between the clusters generated by the transcriptomes and the cell types and cell classes markers. So here we have all the data from the human retina, both foveal and peripheral. And here we have the maps concerning the organoids. The gene markers would be in the Y, and the transcriptome classes would be in the X. And the dots would represent, by their color intensity, they would represent the number of transcripts per cell, and by their size, they would represent the percentage of cells that are expressed in that particular marker. So with this, we were able to detect all the known neuronal cell classes and cell types of functional human retina explants. And for organoids, we also detected that most retinal neuronal cells are present. Some non-neuronal cells are also present. So for example, we were able to see the rods, we were able to detect all three types of cones, we were able to detect horizontal cell types, all the bipolar cell types, the majority of the amacrine cell types. Also, a very interesting observation was that the organoids were recapitulating quite well, or actually very well, the peripheral human retina, but they were weakly correlated to the foveal retina, meaning that our retinal organoids, for now, are mainly recapitulating a periphery of a human retina. So the cell type transcriptome atlas that we created for the functional human retina is also very important to be able to create a map of genes which are associated to retinal diseases in particular cell types. So this is actually the first time that we were able to have transcriptomes from a functionally post-mortem functional human retina. So to do this, we identified different genes that are associated to retinal diseases of the periphery retina. We actually detected, we performed maps for up to 10 retinal diseases. And we identified that many human retinal diseases with genetic association are cell type specific. At this point, because cell types of the adult human retina are also present in our developed organoids, we decided to perform the same disease maps also for organoids and compare them and see if these organoids could be used for a model for species-specific retinal diseases studies. So in this example, for example, I'm showing the disease map associated with retinitis pigmentosa. The genes that are involved in the disease are in the rows, and the cell types are the columns. The intensity of each square indicates the normalized expression of the gene in that specific cell type. And also some of these genes are significantly cell type specific, and are those depicted by a full circle at the end of the row. It is obvious and actually quite interesting how the maps regarding the peripheral human retina and regarding the developed organoids are actually overlapping. And this suggests that our organoids might be a useful model for the study of retinal-associated diseases. So I think that at this point, I'm a little bit over the time, or right with the time. So just to summarize, in this study, we were able to create retinal organoids from human pluripotent stem cells in a high throughput model. And we were able to create multi-layered and light responsive organoids. Also, we were able to create a method to have human retina explants that were light responsive and active. And from these data, we were able to create an atlas of single cell transcriptomes and also a model of diseases which are cell specific to human retina. With this, I would like to thank you all for your attention, and I'm very happy to take questions.
Thank you so much, Martina. That is really amazing. And this is the first time that we have seen, functional developed organoids kind of compared one-to-one to adult human retina.
Yes.
And this is really amazing. And congratulations on this work. And we're really very excited on what's ahead in this research. So before I proceed with lots of questions that we are receiving now in the chat, maybe one thing that is quite curious here. What is next in the research? So now that you have a model of the human retina in an organoid form, what is next?
So I can speak for myself.
Yeah.
I'm not going to speak for other members of the laboratory. Personally, I would like to continue with the electrophysiological characterization of these retinal organoids. Also, I believe a functional and well-characterized organoid would be even more of interest for therapeutical studies. So actually, thanks to Szilard's help, I'm trying out also different kind of stimulations and recordings. So that's for me. Regarding
Cool.
Other people of the lab, there are many studies that are ongoing. We're a very big institute, so let's hope that soon we will be able to talk in another webinar about developments of the...
Cool. Sounds great. Okay, so let's go on with the questions. First, what are the limitations or cons of using organoids, and also for possible developments of future research based on this?
What are the pros and cons?
Yeah.
Okay, that's a long question to answer to. The pros are for sure that these organoids derive from human pluripotent stem cells. So we are talking about species-specific research. Regarding the disease maps, when we want to use models, animal models for these kind of studies, many times the phenotypes are different, or for example, some genes might not be exactly compared. So having a model that comes from human retinas would definitely be a game changer in this kind of studies. And also, if we are talking about tailored research, so a patient that might have a modification or a disease that is genetically driven, we would be able to most likely create, with future developments, maybe create organoids from these lines and then study in a tailored fashion. I'm obviously talking about a future, I'm not talking about the present, but speculating, this would be for sure a pro. Regarding cons, obviously, not very much about organoids is known yet. This is, as you mentioned, one of the first studies where we are showing all of these developments of retinal organoids, but more studies have to be done in order to be sure that we can use this as a model. So that's an obvious con.
But we are just at the beginning,
We are
you
just
know,
at the beginning. Yes,
lots of possibilities. Yes.
I hope I gave a brief response to a very difficult question.
Cool. Thanks a lot. Here we have another question. I think maybe this came earlier in the talk. Did you test any disease mutations in the retinal organoids? I think you showed retinitis pigmentosa. Are there any other disease mutations that you had?
So what we are looking at are genetic maps of genes that are associated to diseases. We do not have yet, or we did not show in this study an organoid that is a disease model. So these are two different things. Of course, it would be very interesting to perform similar experiments on a organoid that is a disease model, but this was not the scope of this study.
Cool, thanks a lot. Okay, next. You see a lot of astrocyte gene expression in organoids compared to the human retina. Do you think this is not concerning for your model?
So actually, this is not really the case in the sense that, for example, Mueller glia cells, which are glia cells of the retina, are very present in the organoids. But I wouldn't necessarily say the same for astrocytes specifically, and not all of the glia or non-neural cell types that are present in the retina of a human retina were also present in the organoids. So, I mean, if this would be a glia-specific kind of study, for sure this would be an interesting characterization. But for what we saw from transcriptomes and quantifications, this was not necessarily the case.
All right. Okay. So maybe we can refer also to the paper for more details.
Of course, yes. Absolutely.
Okay. Now this is about function of the retinal organoid. So until what week is your retinal organoid functional?
So this, yeah. So this also depends on what kind of recordings we are doing. So if we are just looking at spiking activity, this is a big limitation in the sense that we are only looking at ganglion cells. Basically, the ganglion cells are the only cells that are spiking virtually in the retina. So in this case, we have to go back to the developmental slide to see the time course in which the ganglion cells appear and stay, and the peak of presence of the ganglion cells. This is relevant for what concerns, for example, multi-electrode array recordings. If we are doing calcium imaging, so again, activity and electrophysiological recordings, but a completely different kind of model, then the calcium imaging recordings that I showed were done on very developed and mature organoids, which could go from week 30 to week 40 with no problem. And I was always able to see active cells and light responsive cells. We usually do not go over a year of development of the organoids in general.
All right. Okay. Yes. We have another question. So, how might you envision retinal organoids being used to test gene therapies like Luxturna, which replaces a mutated RPE65 gene?
I think that they are very promising, and especially after what we showed here, it's something that is very promising. Of course, it needs more studies, and of course, if one wants to use organoids as a model, many controls have to be done, and also the field is still growing, so you might face some some questions that are obviously because of curiosity. But personally, I think that it's a very good idea, or something that will develop in the future quite quickly. So I would see that as absolutely a model of relevance for these kind of studies.
I would just add also the following, that it opens up the possibility of testing human tissue, because currently, right, many labs are using as an AMD model the mouse, right?
Yes.
Which doesn't have a macula. So your therapy is basically in a way, it's pointless, right? So you need a tissue where you kind of would have the cell types and the arrangement and the properties of the macula. And because of this, the organoids could be very promising.
True. And because, it recapitulates human retina. So this is really very directly related to what human retina would experience. All right. So I think we also have people who are interested to see or to learn more about the protocol, just very briefly on how you developed the organoids.
Of course.
Do you have anything about this? Yes.
Yes, but then I would go and share my screen again.
Yep. So let me just.
Okay. So. You see my screen? Yes. Oops, don't anymore.
And maybe you can also mention about, why it takes so long, and if there is anything to optimize, to make it more developed earlier. Is there anything that can be done there? Yes.
So I don't know if you see the screen, but maybe the three of us are.
Yes, I will turn my video off. Yep.
Okay, so this was actually, I'm very happy that you asked, Mary, thank you, because this is a key point of the paper, but for time's sake, I was not able to include it in the presentation, because we developed this method for high throughput generation of organoids, which was not shown before. This is the first panel is just a summary of how we generate organoids. So how we go from the colony of induced pluripotent stem cells to the actual organoid with its layered structure and everything. So the first step is to generate the aggregates of these iPSCs or pluripotent stem cells, which go by the name of embryoid bodies. That's what EB stands for. So these embryoid bodies are just aggregates to the aggregates of cells, and they can vary greatly in size. They can go basically from 95 to 640 millimeters in diameter, which is a very big variation. So actually the size of these aggregates is very important for the successful outcome of an organoid and for their development in general. So we developed a method to control for this size by limiting their possibility to grow, generating these embryoid bodies in agarose microwell arrays. So this is what you're seeing here. These big circles here are some micro arrays generated in agarose, and we would seed our iPSCs directly there so that they cannot grow indistinctively. Then usually what happens is that these embryoid bodies would be seeded onto matrigel, and this 2D kind of structure would then develop in an emerging retinal structure. So this kind of optic disc-like structure would kind of arise. And what in literature has been shown is that at this point, people would do microdissection manually. And this microdissection takes very long. It takes up to 40 minutes. So what we developed is this different method, which is called checkerboard scraping, in which we are able to dislodge the content of the entire plate in 5 minutes instead of 40. And this also, since we are talking about the content of the whole plate, generates many more organoids. So obviously after 5 weeks from the seeding of the iPSCs, we will have these floating organoids, which I showed also before, and then they would develop. So just to give you an idea of what I just mentioned, here we are showing that the diameter of the embryoid bodies would, its variation would also allow a different percentage of retinal organoids to develop. So if it's too big, the percentage of retinal organoids would go down. And also, it depends on how many cells you are seeding, of course. So we also control for that. Here, we are also comparing what happens with unregulated embryoid bodies and what happens in our system where we have this microwell seeding. In this case, we are able to perform a much higher output and in less time compared to the old method. So we called this method that we created AMAS, so agarose, microwell array, seeding, and scraping, and it's here depicted in green, and it's how many retinal organoids we can get per well. And obviously the comparison is a no-brainer in this case. So this is how we generate organoids in-house, and we talked about the method extensively in the paper if there are more questions that have to be asked.
Amazing. Look
Hope
at that.
this answers your question. Yes, it's crazy.
Yeah, it's really high throughput. Cool.
It's a factory.
Yep. Okay, so I think we are nearing our last question. And this is more related on how to actually record signals from the organoids using the microelectrode array. So there's a question, do we need to cut them? Is it possible to record from photoreceptors?
Okay, so to perform multi-electrode array recordings on the organoids, we have to take into consideration how they are organized. So the organoids are basically spherical, and on their outside part, we have all the photoreceptors. And then the deeper layers are deeper in the tissue. As I mentioned before, if we are doing multi-electrode array recordings, we want to see spiking cells, so we are going to put ganglion cells optimally in contact with the array, with the electrodes, with the chip. And so to do this, yes, it is quite important to dissect the organoids so that the ganglion cells would be in contact with the array. Yes, but it's doable.
Yeah. Szilárd, maybe you can elaborate more. Is there a need to culture these once they're cut, or can you record directly acutely?
So at the moment, we have a protocol where you can record them acutely. We have also tested culturing, but not with great success until now. What one needs to realize that this type of protocol requires a lot of optimization steps. So unlike in brain organoids, where your spiking neurons are technically all over the sphere, the outside of the organoid, here the ganglion cells, as Martina said, they really recapitulate the histology of the human eye or the mouse eye. And the ganglion cells are typically in one particular layer. Mm-hmm. And you need to access this layer. And unlike in vivo, where between this layer, after this layer, you only have the vitreous, and it's an open cavity, so your cells are the surface. In the organoids, this is not yet clearly defined. So yes, you have a ganglion cell layer, but you need to get to it. You need to dissect the additional tissues that might lie on the surface. And at this stage, this is why this is challenging. However, this step is also challenging if one would record, for instance, from mouse retinal MEA. Mm-hmm. The tissue has to be perfectly clean, so that the contact between the cell and the array is optimal.
Also, it's curved, so you have to flatten it as well. Mm-hmm.
Yeah.
All right. And follow-up question to that: is it possible to record from photoreceptors?
On the multi-electrode array? Mm-hmm. Well, indirectly, in the sense that if we would do light stimulation, then the photoreceptors would be the cells that would collect the photons. And then if the circuit is functional and maintained, then we would see spikings from the ganglion cells. So we need a functional circuit to be able to do that.
All right. So that's it. For the questions that we have not answered, we will get to those via email. Thank you so much, Martina, and thank you, Szilárd. And thank you, everyone, for attending our webinar. Please contact us if you have any questions at info@mxwbio.com. You can also refer again to the paper in the links here in the chat, and also the atlas. That's really cool. Please take a look. You can also schedule a call directly with us, with Szilárd, through this Calendly link, and visit our website. And there is a small survey at the end of the webinar. If you want to, have any topics that you want to hear about in the next webinars, please feel free to comment it in our survey. So thanks again, and have a good evening, everyone. At least from Switzerland. Good morning everywhere else. Okay. Yes. Bye.
Thank you. Bye.
Bye-bye.